What is the difference between DNA and chromosomes? A chromosome is in fact a DNA molecule, and in prokaryotes such as bacteria, DNA is basically the same thing as a chromosome. However, in eukaryotic cells, the DNA inside our nuclei is a really big jumble. On the right is a reconstruction of the chromosomes in a yeast cell, and you can see how jumbled up and intertwined the chromosomes are. If you took the DNA in a human cell and stretched it out like a string, that string would stretch for 3 meters. Considering that a typical cell is only 10 microns wide, the DNA has been compressed by a factor of 1:300,000. So you have this big jumble of DNA in the nucleus, but when the cell divides, you want to be able to organize this mess and reliably segregate the DNA so that each daughter cell gets an equal amount of DNA. And that's why the DNA in eukaryotic cells doesn't exist in isolation. It is in fact in complex with a set of proteins called histones, and the DNA is wrapped around the histones. And this complex of DNA and histone proteins organizes itself into more complicated structures, which we will learn about later on in the course. Together, this complex is called chromatin, and it is essential for many biological processes, including cell division and gene expression. And therefore, in eukaryotic cells, the difference between DNA and chromosome is that chromosome is DNA plus other proteins that organize the DNA in the space inside the nucleus. What do the terms haploid and diploid mean? Many organisms, such as bacteria, but also life stages in organisms, such as the red bread mold, Neurospora, as well as our gametes, human gametes, have one of each type of chromosome in the cell. For example, you may have a long chromosome and a short chromosome. And therefore, this cell has 2 types of chromosomes, and this cell is said to have a haploid number of 2. Humans and human gametes have 23 chromosomes, and therefore humans are said to have a haploid number of 23. But other life stages in sexually reproducing organisms, such as humans and flowering plants, namely, for humans and flowering plants, it's the somatic cells, the cells of our body, that have not 1 but 2 of each type of chromosome. And I'm going to show the other homologue of each type of chromosome in yellow. They are called homologues because the two chromosomes of each type, they are similar to each other but not identical. One of the homologs comes from the father, and so is the paternal derived chromosome, and the other homolog comes from the mother and is the maternal derived chromosome. So this diploid cell then has a total of 4 chromosomes. So the haploid number is 2 and the diploid number is 4. How does chromosome number change during the life cycle of a sexually reproducing organism? Such an organism may start life out as a single-celled zygote, and the zygote will be diploid. So it will have 2n chromosomes, where n is the haploid number of the organism. Now, the zygote is going to undergo mitosis, repeated cell division, until you get an adult individual. And we'll talk about it in a bit, but during mitosis, cell division, the DNA is copied, and therefore the number of chromosomes and the amount of DNA does not change in the cell, and therefore this adult is also diploid and has 2n chromosomes. Then this adult is going to undergo meiosis, and one of the most important function of meiosis is to halve the number of chromosomes. So you may get sperm, you may get eggs, but these gametes are going to be haploid, and therefore they will only have n chromosomes, and then these gametes are going to undergo fertilization, and each gamete brings with it an n chromosomes, and therefore in the zygote you end up with 2n chromosomes. And evidently, for each pair of homologous chromosomes in the zygote, one of the pair came from the egg and the other came from the sperm, and that's why this is the paternal and the other one is the maternal homolog of a pair of homologous chromosomes. And therefore, in the life cycle of a sexually reproducing animal, there are 2 key steps that change chromosome number. During fertilization, by definition, since fertilization involves the fusing of the sperm and the egg, there is a doubling of chromosome number from n to 2n, and consequently there is a need for a halving of chromosome number from 2n to n. Therefore, fertilization and meiosis serve opposing roles, so that fertilization doubles the number of chromosomes, and there is a necessity for meiosis to halve the number of chromosomes. And I would like you to imagine what would happen if there was no meiosis to halve the number of chromosomes when you produced gametes, yet you had fertilization that doubles the number of chromosomes. Dividing cells undergo the cell cycle, and there are 4 phases of the cell cycle. The S phase, and the S stands for synthesis. This is when the DNA is replicated in the cell in preparation for cell division. There is a necessary need to replicate the DNA, since if the cell divides without this replication, the daughter cells will have less than their full complement of DNA and would not be able to survive. Then there's the M phase, where the M stands for mitosis or meiosis. And then there are 2 gaps, G1, say gap, and G2, which also stands for gap. And the cell cycle proceeds in the clockwise direction, going from synthesis or doubling of the DNA, copying of the DNA, to G2 or gap phase 2, to mitosis or meiosis, and then the first gap, G1, and then returning back to the S phase. And a further division is that everything other than mitosis is called interphase. Now, the role of meiosis is to halve the number of chromosomes. However, given the constraints of the cell cycle, this happens in a roundabout manner in 2 stages, meiosis 1 and meiosis 2. In meiosis 1, the cell is still in the cell cycle and it undergoes DNA replication in the preceding interphase, in the interphase that precedes the meiosis. And therefore, for a diploid organism, if you started out with 2n chromosomes, where n is the haploid number of chromosomes, you will end up at the start of meiosis, or start of M phase, with 4n chromosomes. And then in meiosis 1, the cell divides in 2, and what that means is that the daughter cells at the end of meiosis 1 have 2n chromosomes once again. And we can look at the number of cells. So if you started with 1 cell, you will end up with 2 cells. At the end of meiosis 1, the cell proceeds straight to meiosis 2, and there is no DNA replication. During meiosis 2, there is cell division, and therefore the number of chromosomes goes down or halves from 2n to n, giving you a haploid gamete. And you started out with 2 cells and you divided each cell into 2. Therefore, you end up with 4 gametes. So every myocyte generates 4 gametes. Let's take a cell with a haploid number of 1, and therefore the cell has 1 type of chromosome, and it's a diploid cell, so therefore there are 2 homologs of this chromosome. Perhaps this is from the mother and the other one is from the father. Now during interphase, during the cell cycle in the interphase, in the S phase, the DNA is going to be copied, and therefore we're going to end up with doubling the number of chromosomes. And therefore this cell had 2 N chromosomes, and now we have 4 N chromosomes, and N is equal to 1. During prophase 1, these chromosomes are going to condense into chromatids, and the 2 copies resulting from DNA replication are called sister chromatids. So these 2 are sister chromatids, and perhaps the one on the left came from this copy of the homologous chromosome. The other one came from this copy of the homologous chromosome. This dot in the middle is the centromere, and this is where the kinetochore is going to form so that the chromosomes can be pulled by the spindle apparatus to the opposite poles of the cell during anaphase. And similar to the paternal chromatids, the maternal chromatids would also be condensed, and the 2 copies would be called sister chromatids. Now, this pair, or each pair of sister chromatids, is called a dyad. And there are 2 dyads here, one from the maternal chromosomes and the other from the paternal chromosomes. Now the 2 dyads are not floating around freely. In fact, in prophase 1, the 2 dyads are attached to each other through a complex of proteins called the synaptonemal complex. And the importance of the synaptonemal complex, or attaching the 2 dyads to each other, is to make sure that during metaphase 1, each dyad aligns on the opposite side of the metaphase plate, so that eventually each daughter cell of meiosis 1, and therefore each gamete in at the end of meiosis 2 receives one of each type of chromosome. The chromatids coming from the paternal and the maternal chromosomes, that is, the chromatids that are not the result of DNA replication, are referred to as non-sister chromatids. This whole unit of 4 chromatids is called a bivalent. And finally, the 4 chromatids together are referred to as a tetrad. With all this terminology out of the way, let's see what happens in Metaphase 1. And keep in mind that the number of chromosomes hasn't changed in Prophase. It's still 4n, and so it will be in Metaphase 1. What happens in Metaphase 1 is that each dyad aligns on one side of the metaphase plate. So that at the end of meiosis 1, one pair of sister chromatids are in one daughter cell, and the other pair of sister chromatids are in the other daughter cell. And since cytokinesis has taken place, the number of chromosomes has reduced from 4n to 2n. And I will not go into all the details of meiosis 2, but the bottom line for meiosis 2 is that the sister chromatids get separated, and therefore 2 gametes get the paternal homologue of the chromosome, while 2 gametes get the maternal homologue of the chromosome. And since there's cytokinesis or cell division during meiosis 2, the number of chromosomes halves, so that you end up with gametes having half the number of chromosomes, n chromosomes, which corresponds to the haploid number for the organism. The final point that I would like to underline here is that meiosis underlies Mendel's law of equal segregation. There is segregation happening, or separation, of the chromosomes into the gametes, since half the gametes get the paternal chromosome and half the gametes get the maternal chromosome. Furthermore, it's clear that after every meiosis, or every myocyte gives 2 gametes that contain the paternal chromosome and 2 gametes that contain the maternal chromosome, and therefore the segregation is equal, so that there are equal numbers of gametes that carry the paternal or the maternal chromosome.